Now we are going to start about the important cytokines. In important cytokines, basically we are going to study about interleukins. First, let's segregate the interleukin word. The inter means between and leukin white blood cell. It means between white blood cells. Basically, the interleukins are way of communication of a messenger between the white blood cells after producing from the macrophages in EKC. The interleukin 1 is causing the fever. Before going to laboratory, having a small mnemonic that is hot T-bond streak. Hot T-bond streak. And you have to give the numbers in sequence, the interleukin 1, 2, 3, 4, 5, 6. The 1 hot fever, the 2 is T means stimulate the T cells. The 3 is going to stimulate bone marrow, hot T bone and streak. ST is nothing, E stands for IgE production and 5 for IgA production, and K is stimulation of acute phase protein production. That is a one of important mnemonic you have to memorize. And secondly, elaborate the first interleukin when you already know what is causes a fever and acute inflammation. It activates the endothelium to express the adhesion molecules, induce a chemokine secretion to recruit the WBCs, and also called osteoclast activating factor. The interleukin 1 is acute phase, interleukin acute cytokine, and it causes basically fever. They activate the endothelium for the adhesion molecule, induce the cytokine secretion for WBCs, and secondly also called osteoclast activating factor. Now come for the interleukin 6. The interleukin 6 also causes fever but stimulate the production of acute phase protein. This is the most important function of interleukin 6 for the production of acute phase proteins. Second, we are having tumor necrosis factor alpha. The tumor necrosis factor alpha is going to activate the endothelium and causes WBC recruitment and vascular leak. And it causes acaxia in the malignancy, maintenance of the granuloma in TB. And also interleukin 1, interleukin 6, and 2 can mediate fever and sepsis. So mean is that if you guys going to an infection and after infection they will be going for sepsis, the main role in sepsis is going to play by tumor necrosis factor alpha. It's also very important point. Interleukin 1, 6 and tumor necrosis factor alpha can mediate fever and sepsis. As you know, and non sepsis means bacteria coat him right. And secondly, there is a maintenance of the granuloma in the TB is also done by tumor necrosis factor alpha. The question for you, what important point? Interleukin 8 is a major chemotactic factor for neutrophils. You can remember that they clean up on S8. Neutrophils are regulated by interleukin 8 to clear the infections. The interleukin 8 are going to clean the infection by neutrophils. Neutrophils go and clear the infection. So interleukin 8 also going to play a role in neutrophil chemotactic. You guys also in the complement system what are C5? Good. The interleukin 12 induces differentiation of T cells into T helper cell 1 and activates natriculosis. The important interleukin 12 and it facilitates the granuloma formation in TB. And that all are secreted by macrophage interleukin 1, 6, tumor necrosis factor alpha, interleukin 8 and interleukin 12. Now we are going to the interleukin that going to secrete by T cells, that is interleukin 2. Or we know it stimulates the T cell. It stimulates the growth of hyper cytotoxic and regulatory T cell natural killer cells. Interleukin 3 are going to support the growth and differentiation of bone marrow stem and function like GM-CSF. They stimulate the proliferation of eosinophils, basophils, neutrophils, and monocytes. What is going to be released by T helper cell 1? It is little bit of summary. If you guys remember the topic differentiation of T cell already we have learned These all the things that topic which thing is released from T cell macrophages and what is secreting from helper 1. It is interferon gamma that is secreted by natural killer cells and T cells in response to antigen or interleukin 12 from macrophages. When the interleukin 12 from the macrophages stimulate the T helper cell 1, they will release interferon gamma. And they will stimulate macrophages to kill phagocytosis to the pathogen. Also initiate or support for the phagocytosis of pathogens. Inhibits differentiation of T helper cell 2. And they induce the immunoglobulin G isotype switching in B cell. Interferon gamma, the first thing they are going to switching of IgG. Secondly, interferon gamma is coming from T helper cell 1. So they are going to inhibit helper cell 2. And secondly, if they are going to macrophages, they will stimulate the phagocytosis of pathogens and increases the MSC expression antigen presentation by also it activates macrophages to induce granuloma formation. So guys, till now we are done with some granuloma formations. First, it is by interferon gamma, by interleukin 12, gamma, interferon, gamma, interleukin 12, and tumor necrotic factor alpha. From T helper cell 2, interleukin 4 are going to release that induce the differentiation of T cells into T helper cell 2 and they promote the growth of B cells, enhances the class switching to IgE and G. And they're too proud to beg for help. It's a mnemonic. It is too proud to beg. Beg means they are going to produce, promote the growth in B cells, IgE and IgG switching, and 4 helping. Interleukin 4 is doing this function that is raising 4 type 2 helper cell. Interleukin 5 promote the growth and differentiation of B cell and has the class switching 5GA. They stimulate the growth and differentiation of eosinophils. I have 5B. 5B means B cell promotion for the growth, IgA formation, and E stand for eosinophil. If you guys correlate the mnemonic and memorize the thing or learn these things within the mnemonics, it will be very easy for you. Interleukin 10 attenuates inflammatory response. They attenuate or decrease expression of MSC class 2 and Th1 cytokines and inhibits the activated macrophage and dendritic cells are secreted by regulatory T cells. You guys already know regulatory T cells are going to regulate, maintain the immune response. So they are anti-inflammatory interleukin cytokines that are interleukin 10 and chemotherapy factor beta. They have a mnemonic that both attenuate the immune system, immune response, both before chemotherapy beta and attenuate 10. This stand for interleukin 10. Interleukin 13 is a promotes IgE production by B cells, induces alternative macrophage activation. They have an interleukin 13 doubly promotes IgE. That's not easy by the mnemonic only. Now we are going to have a topic of respiratory process. It's this mechanism of killing the pathogens. It's also called the oxidative process that involves the activation of phagocyte NADPH oxidase complex in neutrophils and monocytes, and they use the oxygen as a substrate. That oxygen is converting the oxygen radicals, and it's happened by NADPH oxidase. NADPH oxidase. And they plays an important role in immune response, rapid release of reactive oxygen species. NADPH play a role in both the creation and neutralization of ROS. They can neutralize and can create the reactive oxygen species. Myeloperoxidate contain a blue-green heme-containing pigment that gives the sputum its colors. Guys, you know whenever you have a bacterial infection, you go for coughing and the time, therefore, the green or blue colorish sputum is coming, that is due to myeloperoxidase. Due to myeloperoxidase, the sputum color having that bluish green. There's a non-state microbe. Non-state microbe means it's a mnemonic that the NADPH oxidase is going to release superoxide dismutase, and that is going by myeloperoxidase. What is going to happen? The oxygen convert into radical, and that radical going to become a hydrogen peroxide, and the hydrogen peroxide is combined with chloride ion, and then it become hypochlorous acid by the help of myeloperoxidase. So that myeloperoxidase go and give the end stage or destruction of bacteria. Understand? And there is one important point like that. If some person is having the deficiency of NADPH oxidase, so that disease is called chronic granulomatous disease, CGD. In that disease, the body is unable to produce oxygen radical or hydrogen peroxide, but in that time, they can use the hydrogen peroxide that is produced by the bacteria itself and use that hydrogen peroxide molecule to kill that bacteria. But some of the bacteria are catalase positive. They break down the H2O to hydrogen peroxide themselves, so our body can't use that hydrogen peroxide for killing them. So in this disease, in this chronic granulomatous disease, they have the recurrent infections by catalase positive species that are Asorius and Aspergillus. And then the phagocytes of a patient with CGD can utilize hydrogen peroxide generated by invitic organisms and convert into radical oxygen species. The patients are at increased risk for infection by the catalase positive species that are Asorius and Aspergillus, capable for neutralizing their own hydrogen peroxide and leaving phagocytes without radical oxygen species for the fighting infection. The pyrocanine of P. aeruginosa generate ROS to kill competing pathogens and oxidative growth lead to release of lysosomal enzymes. This just an immune response or killing of bacteria by the free radical, and this is already discussed in general pathology free radical topic if you guys remember. Now we are going towards type 1 interferons. The type 1 interferon, why there is an interferon? Because they interfere in the production of RNA and DNA of the viruses. So they are having alpha and the beta and the part of innate host defense. The interferons interfere with both RNA and DNA of a virus to stop and inhibit their growth. The cells infected with the virus synthesize these glycoproteins, which act on the local cells, priming them for the virus defense by downregulating the protein synthesis to resist the potential viral replication and by upregulating MSC expression to facilitate the recognition of infected cells. Also play a major role in activating antitumor immunity. What is happening, for example, in simple words, this is the one cell and these are other cells, B cell like B cell and this is cell, and when bacteria, one viruses come and attack on that cell. In that cell, the interferon molecules are present and they will go to the different cells, these molecules, and give the signal to stop the production of the protein. If the protein production or DNA RNA production will be stopped, then there will be no more growth of the virus inside the cell because the virus needs host protein for their own growth. So they will interfere the growth of viruses and stop the growth of virus and protect the human beings. So we used them previously in our different diseases like chronic hepatitis B viruses, Kaposi sarcoma, Hairy cell leukemia, condyloma acuminata, renal cell carcinoma, malignant melanoma, and multiple sclerosis. But nowadays, due to adverse effects, we are using another substance instead of interferons. They are having the adverse effect of flu-like symptom, depression, neutropenia, myopathy, interferon-induced autoimmunity. It's all about interferon. The basic thing is that they interfere and to stop the growth by interfering to RNA and DNA production of the viruses and inhibit their growth in the host. Now we are going to start up with the cell surface protein. You guys already know about the previous chapter we guys discussed about the T cells, B cells, activation, and all the things. And that there are some type of markers are present. T cell, there is one important receptor, that is T cell receptor, the bind the antigen MHC complex. They're having CD3 that are associated with TCR for signal transaction, and also CD28. 1 and 2, these are needed for the double signal for the activation of the co-stimulatory signal for the activation of T cells, and CD28 going to bind with B7 on APC antigen presenting cells. Helper T cells having CD4 and cytokines and the CD8. And helper T cell also having CD40 ligand that are going to bind with CD40 of macrophages and CD40 of B cells. They are having 6 CR4 and 5, they're the core receptor for HIV. Regulatory T cells, CD4 and CD25, the B cells having the immunoglobulin binds antigens, and they having CD19, 20, 21, the receptor for Epstein-Barr virus, the 21 must be 21 to drink after bar. So CD21 is for Epstein-Barr virus. So CD40 to bind with T cells, MSC class 2, and B7, CD1886 for the co-stimulation. The natural killer cell having CD16 for the antibody antigen activation, complex activation, mediated activation, that will bind to FCFM immunoglobulin G. They have in CD56. That is a marker of natural killer cell. All of you guys know that. And then you have to have in the macrophages CD14, the receptor of PAMs, and CD40 CR5, and MSC2 and a B7 FC, and CD3B receptor that enhance the phagocytosis. This all belongs to macrophages. They are the hematopoietic stem cell, they are having the CD34. The passive versus active immunity in the passive, already that human being getting the pre-formed antibody. The antibody already formed. That most having in babies like IgA in the breast milk and maternal IgG crossing placenta. Also antitoxin and a humanized monoclonal antibody given to patients. But they are having the rapid onset fast, they will like, but they are having that short span of antibody and no memory, and half-life is equal to 3 weeks. But they are the active always depend after the exposure of exogenous antigens. The antigens are given or the patient is infected by some disease, and then its immune system get the action against this organism, and it will be long-lasting protection, giving the memory to that human being. But it's a little bit slower compared to passive immunity. They are natural infection, vaccine, and toxoids. If you read about important point is that combined passive and active immunization can be given for hepatitis B and rabies exposure. And this IVIG and other immune globulin preparation can be administered to provide temporary but specific passive immunity to the target pathogens.